Hello, everyone. Welcome to Nature Nano Talks. I am Fabio Pulizzi. I'm the Chief Editor of Nature Nanotechnology. So our journal was launched in October 2006, and we're celebrating our 15th anniversary. And for this celebration, we have asked some scientists who have significantly contributed to the field in the past, and they still do now. And also they've contributed to the journal. They contributed as authors, as reviewers, and sometimes just as inspiration for us editors and for our readers. We have then therefore started this series that will last for seven webinars from today till the 1st of December. And today we host two particularly significant pioneers of the field, Ben Feringa and Robert Lange. But I will leave the word in a minute to my colleagues, Alberto Moscatelli first and Chiara Pastore afterwards. I just want to remind you that if you want to follow all the other webinars as well, you should please register again for each individual one. But the word goes to you now, Alberto.
Hi, thank you, Fabio. It's my immense honor to introduce to you our distinguished professor Ben Feringa from the University of Groningen in the Netherlands. It's an immense honor because he was one of my scientific heroes as a PhD student in photochemistry. But I'm not going to bore you with the list of honors and prizes he's won. I'll just mention one, the 2016 Nobel Prize in Chemistry for the design and the synthesis of molecular machines. I think that's quite enough as a list. Since we were celebrating the 15 years anniversary of Nature Nanotechnology, I got here the first issue of the journal, and which features molecular machines and molecular motors in the cover page, and inside there is actually a review written by Ben that is here. And I have to say that I read it again yesterday after a long time I had not read it, and it's still a very enjoyable read. And at the end of the talk, I'd like to share with you a link so that you can access also the read-only version of the review, so that you can also enjoy it. He entitled the review "Making Molecular Machine Work." Making molecular machines work. And now, after 15 years later, he'll tell us the sequel, I think, in his talk entitled Dynamic Molecular Nanosystems from Photopharmacology to Smart Materials. The floor is yours, Ben.
Thank you very much, Alberto, and also Fabio, for the kind words. Let me see. Can you see my first slide now?
Yep.
Okay, so then I can start. So once again, thank you so much for the kind words and for making this possible, and for congratulations to Nature Nanotechnology for this 15th anniversary. I think this is not only a fantastic journal, but also it helped enormously to set the stage of the field for many of us, you know. So once again, my great compliments for what you are doing and how you bring the message of the importance of nanotechnology, molecular nanotechnology in my case, to the world. So I will talk about dynamic molecular systems from photopharmacology to smart materials, and share with you a little bit of our journey through this field. But let me put it in the right perspective. I'm a chemist, and we build molecules and materials, and chemistry being a creating science, I think there's tremendous challenges to make sustainable processes and products, reinventing materials, of course, changing the face of our chemical industry, and making drugs not based on trial and error, but on molecular design. And there, nanotechnology and nanoscience will have a tremendous impact on how we are going to do that, because we will address fundamental questions, but also show that there are enormous challenges and perspectives for industry and society. So, in the art of building small, we go back to Richard Feynman. We should not forget that, who said in 1959 there is plenty of room at the bottom. And we all know the story, going from this first computer to the small, tiny, but extremely powerful chips and transistor technology that we have today. So in the top-down approach that was followed over all these years, It was Richard Feynman, already visionary, saying the bottom-up approach will be extremely important. Yeah, look at how Mother Nature assembles all these nano machines and all these nano components in your cells. And I had the privilege 50 years later to get this call from Stockholm together with Fraser Stoddart and Jean-Pierre Sauvage. So I often get the question, why do we need these tiny nano machines? Why do we need this nanotechnology anyway? Or molecular motors, where particularly we are working on. And this was a kind of a similar question that was asked to the Wright brothers more than 100 years ago when they were flying for the first time. So people said, why does mankind need to fly? If God wanted us to fly, he would have given us wings. Nobody would have predicted, of course, that we would build in Boeing 747 or in Europe an Airbus, like you see on this picture, that can carry 400 people, 1,000 kilometers an hour across the ocean, 10,000 miles, you know. We are all admiring the flying bird, absolutely marvelous creature by evolution. But a Boeing or an Airbus is not a bird. The flying principle is different, and the whole machine with its 3.5 million parts is completely artificial. But it does the job remarkably well. Try to carry 400 people, 1,000 kilometers an hour with a bird across the ocean. But we should also be extremely modest, because in all these years, we have not been able to build a bird. We have not been able to build one of the cells of the pigeon. I think I would argue we are not able to build even one of the machineries of a pigeon yet, so there is a long way to go. But what we are extremely good in as chemists, and I'm a chemist, I'm a molecule builder, in building molecules, like ethylene, 160 million tons a year, the basic monomer for plastic, a lot of our plastics, and there's of course a serious issue with plastics, so we have to redesign them and recycle plastics, but that's a different discussion. We can make also very complex molecules like vitamin B12, carbon, hydrogen, nitrogen, oxygen, phosphorus, cobalt, but we produce only 35 tons a year all over the world, as far as I know, because vitamin B12, although being crucial for life to stay healthy, we need only microgram quantities to stay healthy indeed. So hugely complex molecules and very small molecules, very simple molecules, large volumes and smaller volumes. So from simple to complex. So we can build all these materials together with our colleagues in material science, in physics and engineering, in biology, of course, pharmaceutical science, cables and cars, the soaps, the dyes, your clothes, the drugs, the pharmaceuticals that we use every day, all the components of your smartphone. But realize this is the success of the physical sciences. But what we are not very good in is to make something that has dynamic functions. So think about motion at the nanoscale. While Mother Nature does this perfect, think about the rotary motor in your body, which is shown here, the ATPs, or the bio-nanomotors that induce the transport, or the muscle function in your muscles. You know, millions of these motors work together so that you can move, that I can speak to you, that I can see you, that I can walk. All these billions and billions of different motors and machines that make this possible. The ribosome, the protein synthesizing machine, a robot, absolutely fantastic. Bacteria flagella motor, excuse me, that makes it possible that a bacteria can swim in your guts and it go in one direction forward and it can return and go in the opposite direction. And then, of course, the information system in your eye, the optical switching. Millions of these switches, these tiny molecular switches in your eyes make it possible that the opsin, the protein, changes when light comes to the eye, the signal goes to the brain, and we can see. This is where we all started many years ago. This is all nanotechnology in soft materials, in Mother Nature, in living organisms. But we should also realize that it's not easy to compare nanotechnology on soft materials with artificial robots, for instance, in a car manufacturing plant. What you see on this picture here is a robot in a car manufacturing plant, 2 meters in size, absolutely flabbergasting how they build a complete car with these robots, you know. I'm always impressed when I see this working. But then go to Mother Nature, and I mentioned it already, in your body, we have this fantastic ribosome motor that makes the proteins, or at least majority of the proteins in your body, and it's only 24 nanometers in size. And I would say the complexity is certainly of the same or even higher complexity than what you see in a car manufacturing plant. So it's an effect of length scales. It's a different world. The nano world in your body, the nano world compared to the macro world. So it's not easy to compare that, because in the macro world, it's size, mass, inertia, force that play a crucial role. While in your body, the nano machines, it's flexibility, viscosity, intermolecular interactions, interface phenomena, all these kind of things that are typically for the molecular world that you will face. So, and it's not so much about getting motion, because there's a lot of motion in the molecular world, the Brownian motion, of course, the thermal motion. So in the world of low Reynolds numbers, it's much more about how to control motion. And this is the challenge we are all facing. So moving from static to dynamic, mimicking a bit of the functions, you know, that we see in both the macro world and of course in the biological machines in our body, we have to work on these kind of challenges. But there is something else, because we talk a lot about molecules and materials, but going from molecules to dynamic molecular systems and responsive materials, it's not only about the structure. Of course, it's also crucial how we organize these systems, how we amplify the phenomena, all the way from the nanoscale, yeah, a billionfold, all the way to the macro scale. Multi-component, yeah, how we bring different components together and they work together. Interface phenomena, I mentioned it many times. Interface, you know, to make a device or to make an active surface or whatever. Think about the membranes in your body, in your cells. And then different hierarchical levels. At the end, it all boils down to the different functions and tasks that such a molecular system has to do. So bringing nanotechnology to function means that you have not only to think about how you build something, but also what kind of functions, how you organize it, how you interface it, etc., etc. So let me start with switches. And I mentioned it already. We got our inspiration from Mother Nature, 0-1 switches, which you find in your computer, in your transistors, but in particularly, of course, in your eye, where we got the inspiration. So in your eye, you have this beautiful molecule, which is called retinol. You see here the band structure. You can switch it with light to a linear structure. That changes the conformation in the protein, and you get a signal to the brain. And of course, many of these molecules work together to make vision possible, to make an image. So key is that you have switching event under light here. So this is a photochemical switch at the nanoscale, and it should be reversible, of course. And we thought, can we use this to do information storage? But of course, to take this molecule from a natural biological source and then make a computational device or an information storage device is not very viable, because they are not very stable. So we had to build artificial switches. And you see here examples of an artificial switch that we can switch between two states, a left-handed helix and a right-handed helix, M and P, minus and positive. And we switch with 2 wavelengths of light. So yes, we can build, trying to mimic the natural system in your eye, we can build molecular switches, and we can switch between 2 states, 0 and 1, and we put in the chirality here, the handedness, mirror image chirality, because we use it as a non-destructive readout method to avoid that we lose information. And over the years, we have used this to make optical memories. So you see here a typical array of a monolayer of optical switches. We have made switchable protein channels. I will show you an example in a moment. So interface with biological systems. And we use it a lot also to make materials, like, for instance, responsive self-assembled systems like nanotubes, vesicles, capsules, etc. So here is an example where we take a switch, a nano switch triggered by light, and use it to make a nano channel that we can open and close on demand. And for that, we choose the mechanosensitive channel from the coli bacteria in your body. And you see a side view and a top view of this channel protein complex. It's a pentamer, and you see in the open state, you form a 2 to 4 nanometer channel in the membrane. Why is that in the coli bacteria? Because in the bacteria, when the osmotic pressure builds up, suddenly these pores open, these channels, and material can flow out to prevent the bacteria from exploding. So we thought, wouldn't it be cool if we could build in an artificial light switch, a nano switch, and then on command open and close. And this is what we did. So together with our colleagues in biology, we engineered via genomic modification these proteins so that we could build in cysteines, that are these blue dots here in this protein structure. Then we can hook up artificial switches, a light switch. You see here a typical switch, which is called a spiral pyran. You can switch with 2 wavelengths of light between a closed and a so-called open state. And in the red open state, you see charges. The molecule is much more hydrophilic. So we thought charge repulsion, dipole repulsion, and water will be attracted into the pore, and we can open it maybe. And that works beautiful, because what we did is we could incorporate it in a giant vesicle like an artificial cell. We could load it up. We can then irradiate with, and the pore opens, and we can close and open the pore on demand. So we can control now, using this system, flow through a nanopore on demand with an external light signal. So this is really how we engineer, starting with parts from Mother Nature, the protein channel, re-engineer it to build in artificial switches. And we use that, for instance, for controlled drug delivery. But now we put also quite a bit of effort towards self-healing materials. Can you imagine? You make capsules. You take this principle that as soon as you have, for instance, damage, light comes in, the capsule opens, it opens a pore, material flows out, like for instance monomers, and the material heals itself. We moved a little bit further in recent years to open this field of photopharmacology. And here, the idea is that you take a drug, a pharmaceutical, and you activate it on the spot with high precision. So normally when you go to a doctor, you want to have an active medicine. But what we want is to have an inactive one that we can activate with light, precisely there where it's needed. So high spatial temporal control. So for instance, you have an infection somewhere in your body, you take an antibiotic, it does not harm your body, it doesn't harm the bacteria in your guts, but you activate it with a light signal exactly there where it is needed. And we focused on 2 important areas, the bacterial resistance. And although we have facing the COVID crisis, the COVID pandemia, don't forget that the World Health Organization said some time ago, bacterial resistance buildup is one of humanity's ticking time bombs. And we have seriously to do something about it. Cancer treatment, chemotherapy, we all know the nasty side effects of chemotherapy. What can nanotechnology do about it to make smart drugs? And so here you see an example of an antibiotic. This is Cipro, fluoxetine, which is a broad spectrum antibiotic. What we did is, in this structure, we take away the heterocycle, we build in an azobenzene switch, and now we can control on and off stage of the activity of such an antibiotic. So the nice thing is here, we can switch it on from the so-called trans form to the cis form. The cis form is active, and it automatically deactivates, and we can tune that precisely. So it can be off again after 3 hours, as you see in this figure here. It auto inactivates after 3 hours, 6 hours, 24 hours. So the idea is when you activate it on the spot, and when it gets into the body, but when it gets outside, after it has done its job, it auto inactivates and you don't build up resistance. You can see we can take a mask. You see here a Petri disk where we grow cells, bacterial cells, and we can grow a yin-yang figure by controlled irradiation where the antibiotic is on or where the antibiotic is off. And we use kind of similar approaches for controlling anti-tumor activity in compounds to have precise chemotherapy to treat them on spot, for instance, small tumors. That's the idea. We also went a little bit further to think about bacterial communication. Now, I communicate to you by language, we show pictures, etc. Bacteria also communicate and they talk with each other using so-called quorum sensing molecules. And you see here a quorum sensing molecule that we modified again with an azobenzene switch, and we can switch on and off the quorum sensing behavior. And the beauty is now that we can control to some extent the communication between bacteria. And why is this so important? You all know that biofilms are extremely problematic in the hospital. For instance, for surgery implants, when you get this infection, you get these biofilms. They are extremely difficult to handle and to treat. When you have, for instance, an implant, a hip or whatever, you have to go back to the hospital, and sometimes the treatment is very severe. So when we can switch on and off the bacterial communication, that might be a step forward. And of course, this is just a proof of principle, but you can see we can reach 700 times difference in activity, and we were extremely pleased with that. But there is one serious issue, and that is how to get light in the body, to have innocent light. And many groups around the world work on how to make these systems working in the so-called therapeutic window. That is in the red area of the spectrum. As you can see here in this picture, in this figure, you have to be between 650 and 900 nanometers, because then you get deep penetration and the light is innocent, and you might use it in a clinical setting ultimately. And there are now many developments that you can show that you can move towards the red part of the spectrum and the near infrared. And meanwhile, we have shown that you can use this kind of principles now for glutamate transporters, for imaging, but also regulating circadian clocks in the cells, so the rhythm of the cells. And many groups around the world are working on this kind of nanotechnology towards biomedical applications. And once again, I'm extremely happy that the students were able to make antibiotics now that we can switch on and off with red light. So now we really can go to in vivo studies, because now we can use innocent red light to control the biological activity. To put it in a bit bigger perspective of bio-nano future, crossing borders between chemistry, biology, medicine. We all know about genomics and DNA nanotechnology. This is fantastic. I'm not going to discuss about that. Network complexity. We all know all these biological networks. What you see here, ladies and gentlemen, is not the underground or the metro of London, but it is, or Paris or New York. This is just a tiny part of the complex biological network. And if we can control by outside signals, for instance, by light, some of these pathways, by switching on and off, we might have the chance to get a much better control over our biological functions, or learn how they operate. So here nanotechnology can bring also many opportunities. Of course, there is this fantastic field of optogenetics, how to interfere biological functions in the brain. And of course, what I mentioned here, and I showed you a few examples of smart medicine and photopharmacology. Now let me move to molecular motors, then going from switches to molecular motors. And we should realize, we all know that once again, our body is full of motors. A living object, a living system has all these machines to make it possible that we can walk, we can speak, we can have all these movements. It pushes constantly a living system out of equilibrium. We should not forget that. And this is the big challenge for the science field, how to push things out of equilibrium. And you see here one of the most fascinating motors, which is the ATPase rotary motor. You see it spinning here. It goes in one direction. There are millions and billions of these motors in your body that make the ATP the fuel. So what were the fundamental challenges for us to design nanomotors? How to control rotary motion and how to control left or right. Because think of your car. If there's no preference for going backward or forward, there's equal probability you wouldn't get anywhere. So how do we control chirality? How do we control, directionality? It's through chirality. We all know that chirality can give the bias. And the most beautiful example, which was called the Mona Lisa of modern science by Nature, is the right-handed DNA helix, but also the left-handed forms of the amino acids, the right-handed forms of our sugar. So what we did is we built in this chirality, and in fact we built two chiral elements, and we built this tiny motor, 1 nanometer in size. You see it spinning here. How can it spin? Because it gets the energy from the light. It's powered by light. So it's a nano-sized motor powered by light. Why do we call it a motor? It has controlled motion. It consumes energy. It is directional movement. We can move it clockwise or counterclockwise, and it is a repetitive process. It goes through 4 steps, 2 photochemical steps, which are extremely fast, like in your eye, the process of vision, and 2 thermal helix inversion steps, which are the slow steps. But together they add up to a 360-degree rotary cycle. Now you might ask me, how fast was your motor? Initially, our motor was rotating once an hour. That's not much of a motor. But meanwhile, we have made dozens and dozens of different motors, and we can rotate them now at 10 million rotations per second, and everything in between, from minutes to seconds to microseconds for specific applications. So more recently, we are towards molecular machines. We try to couple motion. And here my students got much inspiration from the moon going around the Earth. We all know that we always see the moon from the same side. That is so-called concerted or synchronized motion. And you see here two rotating elements that are connected, and the motion is synchronized. And this was a major step for us in recent years to go to couple motion and to amplify motion into real machine type function. So here you see a rotor part. It's a light-driven motor. It rotates. All the components rotate, but it's very well controlled because it's like the moon. They have synchronized motion. So we have put these motors on surfaces, and we have used them to rotate micro objects. I will show you an example. We have used them for macromolecules, for polymers, control materials, and we have showed movement at a single molecular level. So at different length scales, different types of dynamics. So the question was, can we go from rotary to translational motion? And for that purpose, we designed our nano car. We took inspiration from this beautiful solar panel powered cars, 2 meters in size. And my students designed a 4-wheel drive nano car. So a 4-wheel drive, the 4 wheels are the rotating motors, and it's 1 billion times smaller. And you see it moving here over a surface in this small movie. But realize, again, this is how we go from rotational motion to translational motion. That was the design. And you should realize here that we had to build this complex molecule, but the nano world is different. It's not the movement like the movement of your car. This is more likely the movement. It's a stepping movement over a surface. This is how your muscles move. The protein motors, they walk, like this machine here. They walk over a surface, over a filament in your muscles. So here is a modern electric car, and you see in this vial here our nano cars. This tiny amount of powder is pure 1 billion times 1 billion identical nano cars, because they are only 2 nanometers in size. So I would claim that our car manufacturing plant is not so bad after all. So let me show you a few more examples how we design these systems. For instance, we can amplify motion. And what we did is, for instance, we took these motors and we put them into a liquid crystal material. And you all know these liquid crystals in your displays, your smartphones, etc. These are these rod-like molecules. But when you put in a chiral dopant, like our molecular motor, you get a helix organization. And due to this motor function, we can now contract and expand the helix and get these color pixels. So we can make different color pixels. What is more exciting, though, is when my students put these motors in a thin film of liquid crystal material. And this is what you see here. The surface with a thin film of liquid crystal material with a tiny glass rod swimming on top of it. And when now we use simply light, we don't touch it, it starts autonomously to spin. The whole surface changes. You see this soft surface changes its structure. The color changes, I mentioned that before, but you see also the object rotating in a unidirectional sense. So we amplify the motion through many length scales, and we can control the rotation of the change of a surface and the rotation of a micro object. And this is the mechanism, we think, that it winds and unwinds the helical organization. This is amplified then to change in the surface properties. Now, with that in hand, we build polymers. And recently what we did is we put these motors in liquid crystal polymers. And now I don't want to go into details, but we have now polymers that we can bend. We can make them walking over a surface with small steps, and we can make them twisting right-handed and left-handed. So again, taking advantage of this amplification mechanism, the motion of the motor, the change in chirality, and then the control in a polymer, we can get these different types of motions, which might just be a stepping stone ultimately to soft robotics. Now you might wonder, this is amplification. What type of mechanisms are there for transmit this motion? And what we did, for instance, we took inspiration from Jean-Marie Lane's work. And Jean-Marie Lane, the father of supramolecular chemistry, he showed this beautiful double helicates. And we thought, what if we take this helicates and connect it to our motors? And can we rotate under the influence of light and go from left-handed helices to right-handed helices in these supramolecular systems? And that worked really beautiful. So we can have now a system where we can control helicity, we can push it out of equilibrium, and we can change from monomers to oligomers, but in particularly, we can go from right-handed helices to left-handed helices. So this is another way to control assembly at the nanoscale and to amplify, and to push a system out of equilibrium. Now, you might ask, what about surfaces? And I'm from the Netherlands, and we tried to build a windmill. And what you see here schematically is a motor with two legs that we assemble on gold, and we can assemble it now, meanwhile, on many other surfaces. And this was a long journey. It took us 8 to 10 years to make these systems, assemble them as monolayers on surfaces via covalent bonds, and then we can irradiate with light, and we have now them spinning in a unidirectional sense. So coming from the Netherlands, once again, we like windmills. And you see here the windmills of our ancestors 500 years ago. We built this nano windmill park. One monolayer of these tiny windmills, 2 nanometers in size. They rotate all in the same direction, and they make responsive surfaces. So we can make them moving on surfaces with different orientations. You see here another way of orienting and connecting to surfaces. We can control wettability, we can control surface thickness, and this is now a hot area where we make responsive materials and responsive surfaces. And again, many groups in the world work on this, maybe not only with our motors, but once you know how to make, using nanotechnology, responsive surfaces, monolayers, etc., you can think about self-cleaning and self-repairing materials. So in the near future, we'll probably have self-cleaning windows, self-cleaning solar panels, or your car with a coating that can clean itself, or maybe when there is a scratch, that it repairs itself because the surface can be dynamic under the influence of light or an external factor. Now, in the last 2 examples I want to share with you is 3D confinement, and make an artificial muscle. Let me start with 3D confinement, because people ask me all the time, this is nice that you can put it in surfaces, but what about 3D? And we all know that metal organic frameworks is one of the hot areas in chemistry and material science, these porous materials where you have layers and pillars. And what we did is we built these motors now in yellow, you can see them here, so that they can be the pillars in these MOFs. And if you have enough space, they can rotate freely, and you can make now the responsive porous materials where we have them making dynamic. And so when we published this in Nature Nanotechnology, we could demonstrate indeed that we have these 3D crystalline materials, or these frameworks, where all the pillars are motors, and when you irradiate with light, you can make this responsive. And the beauty is now that you can use this in different ways. So we make also the layers, for instance, like antennas with palladium porphyrin complexes that can pick up the light, they transfer it to the motors, and they rotate. Or you can make covalent aromatic frameworks, a different type of very robust materials that are porous. And we use these materials now as smart membranes, responsive catalysts, or release systems. So this offers up a lot of opportunities to use them for membranes, or maybe for separation technology, etc. Now, in my final example, I will share with you another adventure, and that is how to go into water. Because if we want to interface these nano machines with biological systems, or mimic some of these biological functions, we have to go into water. And we focused here on muscles. And you see a muscle movement. We don't know what muscles are. We use them all the time. And you see that in these muscles, you have these filaments with these motors. And these motors walk over these filaments and make these filaments glide. So it's amplification of collective motion from the nanoscale to macroscopic dimensions. Amplification, because millions of these motors work together. Now, Sauvage was the first to make a synthetic type of muscle, but we thought, will it be possible that we use our motors and then use not polymers or so, but can cooperative, non-covalent interactions of small molecules support an amplification all the way from the nanoscale to the macro scale, and then in water. So we make these motors that are amplifiers, like detergents or soaps. They indeed assemble in water. You see these nice structures, these 5 to 6 nanometer type fibers. And then we use the trick, calcium, and calcium binds to the carboxylates and assembles these fibers into nice muscles. And what you see here in the lower corner, you see this alignment of all these fibers. And now we can draw a muscle, an artificial muscle, and you see it in this cuvette. And it is moving. When we switch on the light, you see this muscle moving. And ladies and gentlemen, we can move it in both directions. The muscle can pick up a piece of paper, and what you see moving here is 95% water and only 5% of our molecular motor that was engineered at the nanoscale to make an artificial muscle that can amplify all the way from nano to macroscopic dimension. Of course, it's still soft and it's a primitive system, but this might be a stepping stone to soft robotics in the future. So I shared with you today how to go from molecules to molecular machines, nanoscale responsive materials. We start with Mother Nature. We take inspiration from the beauty of the biological system, and we try to make smart drugs, self-cleaning surfaces, or self-healing maybe, soft robotics, or responsive materials. These are prospects for the future, but once you are able to design dynamic functions at the nanoscale and taking advantage of amplification, synchronization, these things is possible. It would not have been possible, not without the support and the great work of my students. You see my team here. These are my young heroes. They make it possible. Their hard work and creativity. Of course, the funding agencies, but in particular, I have to thank the young members, the students, the team, the young researchers for all their efforts. And finally, a message to all of you in the art of building small and moving at the nanoscale. Remember the words of Leonardo da Vinci. When nature finishes producing its own species, man begins with the help of nature to create an infinity of species. And especially for the younger people in the audience, imagine the unimaginable. I thank you for your attention.
Thank you very much, Ben. It's been a enlightening and inspirational talk for sure. I have a few questions from the audience that have come up. I'll read them out loud to you so you can answer. One is about something you just touched upon, which is antibiotic resistance.
Yes.
If you can explain a little bit more on how nanotechnology or photopharmacology can address that issue briefly again.
Yeah, I'm pleased to do that. You know, when you build in these switches, you can switch between two states. One of the states is an off state. The antibiotic is not active, and the other state is an on state. So when you activate the antibiotic exactly there where it's needed, on the spot, yeah, say you have an infection somewhere in your body and you activate it there, it's not active in other parts, and you can temporarily activate it. And when then it switches off automatically because it switches back, yeah, to the off state, because we can engineer it so that it goes back in a couple of hours or in 24 hours, it's off. It does not interfere with the bacterial growth. We proved that in the laboratory. The bacteria grow normal because it's not an antibiotic anymore. Then we can switch it on again, yeah? And we have done it several times. So that's the whole idea, to make smart drugs that you can, of course, it will not solve all the problems in the drug design, but for specific applications like treatment of tumors without all the nasty side effects of chemotherapy, or trying to find innovative ways for bacterial resistance buildup, I think we can use this kind of smart approach, taking advantage of nanotechnology.
Another question related as well to photopharmacology is, can we use photopharmacology for natural product drug delivery?
Yeah, maybe, because you can also think about building in these systems into delivery systems. I showed you these capsules, you know, that you could open and close. You can build in a drug, but you could also say, I can control the penetration to a membrane or inside the cell of a drug. We have not done that, but it could well be viable. I'm not saying people work on drug delivery all the time, and to take advantage of smart methods to get it in and out, yeah, who knows? So I think there is opportunities. We focus more on the activation of the drugs. Yeah?
And I have also a question very related to this. Has any of your photopharmacology approach been approved by any regulatory agency, the FDA?
No, we have not got to that stage yet. We have done the first in vivo experiments. We have done a lot of in vitro experiments. We have also patented a number of these systems, yeah, for specific applications. But we are not at the stage yet that we have enough in vivo data to have extensive in vivo studies, etc. But we are more than happy to team up also with pharmaceutical companies, you know, to work on this.
Mm-hmm. And I would like you to tell us a little bit more on what inspired you to make these crowded alkenes at the very beginning. At the very beginning, just.
Now that goes all back to a very fundamental question. You probably know when you have biaerials, yeah, you can have chiral helical structures because of hindered rotation around a single bond. And the whole idea was, can you make hindered rotation around the double bond? Now you will say, of course, because you don't have free rotation around the double bond. In your eye, you have to put light there because you break temporarily the double bond, then you can rotate. So we thought, if we make a system that is helical, it is overcrowded alkenes, are they chiral? Can they make a chiral structure, like a helix structure or so? Yeah, and that turned out to work well. And then we thought, okay, if we put light on it, can we break temporarily the bond and control the rotation? And due to the chirality, we can control the directionality of the rotation. So that is the whole basic idea, yeah. It's very fundamental stereochemistry.
And there is another question here about this your molecular motors. Can these molecular motors be introduced in 2D in biomaterial surfaces or other types of biomaterials, for example, in nitrogens, to stimulate cells as well?
Absolutely, that's a very good question. We published a couple of papers. I had no time to discuss this today, but we have made some motors into gels, and we can control the gel properties, you know, the aggregation properties in gels. And we are now currently using these motors also, especially in membranes, you know, to look at the porosity of membranes in the aggregation. Also, James Tour in Texas, for instance, and other groups are working on this. He had a beautiful paper where he used the motors to drill holes in cancer cells. So there is quite a bit ongoing to introduce these dynamic functions in the context of biological systems, you know, both for making active. I can show you one. I didn't show it today, but what we did is, for instance, we put, I have the slide somewhere, but let me briefly explain. We put these motors on surfaces as I showed you today. And then we grow stem cells on top of it. Now, stem cells can differentiate, as you know that. This is a hot area. And so we can now, by using the motor to rotate, control differentiation of stem cells through the protein layer that is on top of the motor, because stem cells are very sensitive to mechanical effects. So we make responsive surfaces and grow stem cells. So this is the interface with the biological system. And I think there's quite a few opportunities there. Yes.
Last question, probably. So light penetration in deep tissues in photopharmacology might be a limitation there, right? So are there ways to address this challenge, for example, using other triggering mechanisms such as ultrasound waves, magnetic?
No, you're absolutely right. Light, of course, initially we will use it for not for organs that are not deep lying, although the people in the hospital tell me don't worry too much about the light, because with this light, how do you call this, fibers, they can get light everywhere in your body. But I think you are absolutely right. There are other trigger mechanisms, you know, think about ultrasound and so, I think this is absolutely true. This is just a lot of possibilities for the future.
Okay, this was the last question, and thank you very much for your talk and your time, and I think the audience appreciated very much. Last thing I want to do is to share with everyone the link so that they can access your review
Okay.
from 2006.
Yeah. Thank you for mentioning it. That was indeed I enjoyed.
So that's the link.
Thank you very much for the opportunity, and much success with the symposium. And once again, congratulations.
Thank you very much. Thank you very much, Ben. I will leave now the stage to Chiara, my colleague, Chiara Pastore, who will introduce you our second speaker. Chiara.
Thank you very much, Alberto. Hi, everyone. I'm Chiara Pastore from Nature Nanotechnology. So thank you very much, Ben, for this really, really fascinating talk. I really enjoyed it. Just one note for everyone, please use the chat, the question box to send us questions, because we will have a Q&A section at the end of the talk as well. So it is really, really a real privilege to have with us today our second speaker, Professor Robert Langer. He doesn't really need much of an introduction, as I know that everyone knows him, so I will keep the introduction very short. Professor Langer is the, here he is. Hi, hello. Hi. He is the David Koch Institute professor at MIT, where he leads one of the biggest and the most prolific bioengineering labs in the planet. He started off as a chemical engineer at Cornell University, and then moved to Boston, MIT, and Harvard to pursue his graduate and postgraduate studies. He is still an extremely and very active scientist, and he works at the interface between biotechnology and material science, specifically focusing on drug delivery and tissue engineering. He has been honored with a really long list of prizes that's way too long to list here. He has always coupled his scientific efforts with a really strong entrepreneurial attitude, and indeed he has filed over 1,000 patents. Some of these have been licensed to pharmaceutical companies. Some of these have actually reached the clinic and are currently helping patients. He's also founded and co-founded several biotech pharma companies, and the most famous of which is probably Moderna that nowadays I'm sure everyone knows. So it's a real honor to have him with us today, kicking off this 15th anniversary webinar series. And today he's going to share with us his view on how nanotechnology has helped opening a way to new strategies for disease treatment. So thanks so much, and I'll leave the floor to you, Bob. Thank you very much for being with us.
Well, thank you so much. Can you hear me okay?
We can, yes.
Thank you. So anyhow, it's an honor for me to be able to present that to you today. Could I have the first slide, please? Thank you. So what I'd like to talk to you about is from our journey and actually others as well, from microtechnology to nanotechnology and new ways to discover and deliver medicine to treat diseases. So first, on the next slide, what I just thought at a very high level, people might ask, well, why is nanotechnology important at all in medicine? And there are a number of reasons why. I mean, that I think interesting advantages. First, that you don't, you can inject things into the bloodstream and it won't clog it up and create problems. Two, as I'll go over, you can get a lot of drug into cells, and in some cases specifically. Also, there's, and this may be obvious, there's much greater surface area, and that advantage, translates in sometimes like diagnostics and imaging to a much greater amplification. And then you can even create new surface patterns, which could be useful, say, in areas that we've also done a lot of work in, like tissue engineering. I'm not talking about that very much today, but I thought it's worth mentioning. Just to give you areas of impact on the next slide. If we could, yeah. So you can, some of the ways that nanotechnology has already really made, I think, a very big impact are enhanced imaging, more sensitive diagnostics. There's a company that Michael Sema and myself and Tyler Jackson, Ralph Weisleiter started called T2 Biosystems, and this is a way of using amplifying signals like from MRI that can allow you to normally get a diagnosis that might take, say, 5 or 6 days. Now you can get it in a couple of hours. Targeted drug delivery is a huge area, and I'm going to mention that. Also, there's been enormous amount of talk about new, in the biological and medical community about new drugs like siRNA, messenger RNA, DNA, CRISPR, and all of these, drug delivery, using nanotechnology or microtechnology is key. And then finally, as I mentioned, the idea that you can help guide the creation of new tissues. But mostly what I want to talk about today is I want to get across that all these areas are important, and maybe in the question period I could answer things on them if it's helpful. But I thought I'd start by focusing on and cover most of what I want to do today is to talk about nanotechnology drug delivery. And if we could go to the next slide, I just wanted to highlight really some of the things that this is enabled to happen. First, that nanoparticles can be taken up by mammalian cells, so whatever you put in them provides a way to deliver them. And I would say the whole area of mRNA vaccines, which of course has changed the world, and I'll talk about that more too. If you didn't have nanoparticles, you wouldn't have these vaccines. You absolutely need them to protect and deliver the messenger RNA. And also, many other potential applications are enabled, like drug transport through nanopores and nanosystems. So I thought I'd start and tell you how I got involved. It was a very unusual way. I actually got involved myself starting in 1974. And I was just finished my chemical engineering degree, and all my friends went to oil companies. And I wasn't that excited about that. And I was fortunate that Judah Folkman, a very well-known surgeon, asked me to join his lab as a postdoc. And he had this idea that if you could stop blood vessels, you could stop cancer. And so my job, he said, was to prove he was right. It turned out to be a very controversial area. He was widely criticized, but what my job was to prove he was right, and in so doing, isolate the first substances that could stop blood vessels from growing. And one of the key problems in this area was that there was no way to study the phenomena of blood vessel growth. There was no bioassay, so we had to invent one. And the way we thought about this was that you could take the eye of an animal that didn't have blood vessels normally, cornea, and you could put certain types of tumors in. And that would actually cause blood vessels to grow. And now you have, it's almost like a graph. You could watch the blood vessels grow from the bottom of the eye towards the tumor. And notice there's no background blood vessels in this, which is not true for almost any other part of the body. So at any rate, our idea was that this might take a while, a few weeks or if not months. And what we would do is put like a polymer or lipid or some delivery system right next to it that could deliver large molecules. By the way, I should point out that all these molecules we were studying were large, like peptides, proteins, nucleic acids. But when and actually at this time in 1974, there was only one company, to my knowledge, that was working in this area at all, or at least to any appreciable degree. And that was a company called ALZA. This was in 1974. Dr. Folkman was an advisor to them. And they had many famous people on their advisory board, like Paul Flory, who would win a Nobel Prize in chemistry. But they all told him that you couldn't do this, that you couldn't deliver large molecules from these little vehicles. And the next slide is an example of what they said, was that large molecules just can't diffuse through solid materials. And the literature said something similar, which is shown on the next slide, that at least for polymers, that strictly restricted to small molecules. But it was absolutely essential, if we were going to solve this angiogenesis problem, that we have a way to deliver the large molecules. So I actually spent 2 years in the laboratory experimenting with different techniques, and I actually failed over and over again. I found over 200 different ways to get this to not work. But eventually, by studying different polymer and lipid designs and and studying different materials and so forth, we were able to make little microspheres or nanospheres. And just to show you a picture of one on the next slide, what you can see is on the left-hand side, you see a picture of one of these, and on the right-hand side, it's cut in half. And we published in the British journal Nature, next slide, the fact that you could use this approach to deliver molecules of literally any size. And in fact, this was also the first time, to my knowledge, that anyone showed that you could also deliver nucleic acids like DNA or messenger RNA for any period of time. We actually showed in this Nature paper that you could deliver it for up to nucleic acids for up to 40 days. Nonetheless, when I first presented this work, it was met with enormous skepticism. I remember being a young guy, 27 years old, and I was asked to give this talk to this very distinguished audience of material scientists and engineers. And I thought I gave a decent talk. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium and a whole bunch of them came up to me and said, "We don't believe anything you just said." Next slide. They said the large molecules can't slowly diffuse through solid materials any more than, like I say, any of us could walk through a brick wall. And also we were using organic solvents, to help formulate these. And they said that would destroy whatever we'd put in, peptides, proteins, or nucleic acids. And actually things went downhill from there, if that was possible, and it was. I actually wanted to get grants for the work I did. So I wrote a number of grants to the National Institutes of Health and other places. My first 9 were soundly rejected. I remember one of them came back because I was doing cancer work. One of them came back and said, like, Dr. Langer's an engineer. He knows nothing about biology and even less about oncology. And I felt like even I know enough about math to know that that couldn't be true. But at any rate, it didn't matter. I wasn't the one that was calling the shots. So I didn't get those grants, and that was difficult. And I liked very much being a postdoc with Dr. Folkman in the hospital. It was great being an engineer and having all these biologists around. I learned so much. But a lot of my friends said it's probably not so good to be a postdoc forever. So I started to apply to chemical engineering faculty positions. And I applied all over, and not a single chemical engineering department would hire me. They all felt like this biological. I got some interviews, but then, and I hope I didn't interview too badly, but nonetheless, what they said at the end was that this biological work that I'm doing doesn't make any sense. And for an engineering department, I should be doing things like more mathematical modeling or oil work or whatever, so forth. So I didn't get any jobs. But what happened is, and I didn't get any jobs in chemical engineering. What happened is Dr. Folkman knew Nevin Scrimshaw, who was also on that advisory board, and Dr. Scrimshaw, who was head of the nutrition department at MIT, offered me a job. But Dr. Scrimshaw was kind of like what I'd call a benevolent dictator kind of department head, and he liked me, so he offered me a job, but he didn't bother to ask anybody else what they thought in the department. And that might have still been okay, except for one thing, which was that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, on the next slide, this is from Michael Marletta, who was a member of the National Academy of Sciences and chair of Berkeley's chemistry department at one point, which is a superb chemistry department. He was giving a lecture a few years ago when I won an award about what it was like to be with me in 1979 or 1980. And he's and this is just a quote from that lecture he gave. He said, one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist said, quizzing us while smoking a cigar. He said, when the older scientists heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that wasn't so good. Nonetheless, I'm still at MIT, so things eventually worked out, but it wasn't easy in the beginning. One of the things that we did is we kept, as I mentioned, publish in Nature that you could, developed the delivery systems, and that enabled us to study this phenomenon of angiogenesis, which people, like I say, were very skeptical about. And in 1976, and I want to give a warning before I show this next slide because it's bloody, but there's no other way to show how blood vessels grow. But in 1976, we did a study and published it in Science, where we studied literally 50 to 100 fractions of angiogenesis inhibitors, and almost every one did not work except one. But what we see on the left, and we did probably over 1,000 eyes like this, different fractions that didn't work or sham controls, but what you see is about 8-9 weeks into the experiment, the sheet of blood vessels growing over the little drug delivery vehicle on its way to the tumor. And if you looked at an eye like this just 3 weeks later, it would be three-dimensional out of the orbit of the eye, it would kill. But what was striking is the right-hand side, where this is a study we repeated 21 times. It was the one fraction that worked, and notice that the vessels are lower, they're sparser, they avoid the drug delivery vehicle, and interestingly, 60% of the time, these tumors never grew. Now, we published this in 1976. It still took another 28 years and literally billions of dollars by different companies like Genentech, and really great work by scientists of those companies like Napoleon Ferrara, to really get FDA approval. It was just enormous. I mean, it's always a challenge doing that, but they did that. But and like I say, in this case, it took 28 years building on this work and other things that Dr. Folkman and I and others had done. But what happened 28 years after this, next slide, was striking. One after the next angiogenesis inhibitor would get approved by FDA. Avastin, which is the first of these, it's probably a number 2 or 3 best-selling biotech drug in history. And not only are these approved for all kinds of cancers affecting, tens, if not hundreds of millions of people, but they're also the main treatments for diseases, blindness, diseases of the back of the eye, with like macular degeneration and diabetic retinopathy. Before this, in fact, there were no pharmacologic treatments for those diseases of blindness. But now drugs like Lucentis and Eylea are blockbuster drugs, which are widely used. Also, Dr. Folkman, as we started doing the drug, I'm going to go back to the drug delivery work, said to me one day, said, Bob, we should file for a patent. That might seem very common today, especially for people who know me, or our lab. But then in the 1970s, I didn't have any patents. And Boston Children's Hospital, where I was working, and it's one of the best hospitals in the world, they didn't have any patents either. So we filed for a patent. And 5 years in a row, the patent examiner rejected it. And so the head of the tech transfer office at the hospital came to see me one day, said, Bob, you are wasting an enormous amount of money. This is never going to get allowed. Just quit. But I don't like to quit. So I started to think, how could we get the patent examiner to allow this patent, legally, of course, and science really wasn't working. But as I told you all in the beginning, when we started this work, everybody told us it was impossible, it would never work. And we wondered if anybody ever wrote that down. So in 1982, I did what's called a science citation search, looking back at our 1976 Nature paper to see who cited it and what they said. And I found some very interesting papers. It's always interesting to see what people write about you, but I just wanted to highlight one of those on the next slide. And this was from 5 of the top materials people in the world. And they were describing, it turned out in 1979, this whole field of drug delivery. And they said generally the agent is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules like proteins could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising. So surprising, that's a really good word for a patent examiner. Surprising results that clearly demonstrate the opposite. So I showed that to the head of the tech transfer office at Children's Hospital. He flew to Washington to see the patent examiner, and the patent examiner said, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these people that they really wrote it. So I was this young guy, I wrote these 5, very well-known scientists, and they were all nice enough to write me back that they really wrote it. And with that, the patent examiner allowed the patent. It was a very broad patent. Next slide. And and controlled release of macromolecules. And that would ultimately lead to companies that I helped start, to companies that gave us grants, and we would train how to do this, and the hospital would license the patents. And today, based on this, there's all kinds of products affecting hundreds of millions of people every year. I'll just show you pictures on the next slide of these. But if you go to the next slide, what you'll see are, I mean, just a few examples. I won't go through all of these, but there are little microspheres like Lupron Depot, which you see in the middle right of the slide. That could deliver luteinizing hormone, releasing hormones that could, and normally, if you tried to swallow them, it wouldn't work. If you inject it, it doesn't work either because it's destroyed right away. But here they're delivered continuously originally for 1 month. Later, Takeda, which developed it, did it for 6 months. But there's many, many others. There's new ways of treating like opioid addiction, where you see Vivitrol. That's one of the main ways of treating opioid addiction today. That lasts for 2 weeks. Risperidol Consta, for a month. Risperidol Consta is a way of treating schizophrenia and all kinds of other things, drug-eluting stents and so forth. So then we wanted to push this even further. Most of these systems are injected underneath the skin and deliver the drug for a while. But and you could do one of three things with drug delivery, right? You could do systemic delivery, you could do local delivery. Drug-eluting stent and the gliadel wafer are very good examples of that. Drug-eluting stent is physically placed in a blood vessel, and you're delivering very high local concentrations, which actually is probably the main way to treat heart disease today. You're locally delivering Taxol. Gliadel wafer is a treatment based on our work with Henry Brem, which you put in the brain to treat brain cancer. The third is targeted. The drug finds its way to a target. And the next slide basically shows that people have used single molecules like Humira, but if you could actually put the drugs in a nanoparticle, you get a much higher payload. So if you think about a hockey, right, you're trying to get into a goal, well, you'll have a much better chance if you're shooting a lot of pucks into the goal than if you just shoot one. And so that was what we were thinking about. And we've made this wish list. We did this collaboration with Vladimir Torchelin. Ruxandra Graf in our lab at the time led this work. And we had this wish list. Next slide. We wanted a high encapsulation amount, high efficiency, that they could be freeze dried, not aggregate, be degradable, be small, less than 200 nanometers, so they could get into a cell and not be rapidly cleared. And what we did is shown on the next slide, that we could make coatings with polyethylene glycol, which could actually provide a disguise. And I should point out that there are a number of groups that would, we collaborated with Vladimir Torchelin, who was one of the groups that did some of the early work on this, but there's others too, who had done work with these in different manifestations, like Bob Davis, Leaf Wang, and Dr. Katoka from Japan. He did my cells. But anyway, we wanted to use this disguise with polyethylene glycol because it would look like that there'd be a lot of water. It would attract a lot of water, and if you see water, the macrophages, which normally clear these particles, will probably clear them much more slowly. That was the idea. We published this in Science. The next slide just shows that they are in fact nanoparticles. This uses quasi-elastic light scattering, and the next slide really just shows how effectively they work. We put an orange dye in them and saw how long they would last when we incubate them with macrophages. If you don't have the PEG, you can see in an hour, look at all the little orange dots in the cell. But when you do put the PEG, like a 5,000 molecular weight one or a 20,000 one, you can see that you get a lot less taken up. And then Avi Dome was a postdoc with me and then came back for a sabbatical, and he actually made conjugates with citric acid attached to these and had a triple PEG. And there you can see on the bottom right with the triple chain, there's actually no uptake. And then Omid Farrokhzad came to our lab, next slide, and started putting targeting molecules on these. And I thought I'd just show you a video that Nova, the TV show, did on all this nanotechnology work we did. Again, this is just to explain it much better than I'm explaining it. If we could go to the next slide, which is a video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So I should point out, a lot of clinicians I work with tell me it doesn't blow the cell up quite like that. So at any rate, we initially did the work. And the exciting thing, of course, is that you could deliver small molecules like this. You could deliver genetic therapy molecules like siRNA or mRNA like this. What I was going to say is the clinicians I work with tell me it doesn't really blow the cell up like that. At any rate, we've used both polymers and lipids. We started a company called Bind, and I want to point out that we've had both success and failure commercially when we've tried to do this. Bind, we moved it into the clinic. This was based on Omid Faraka's work, but but one of the clinical trials didn't do as well as we had hoped. Not that it was bad, but it just didn't do as well as we hoped. And immunotherapy came along, and was a better way to treat the people with that type of cancer. So Bind, we took a loan, and unfortunately, the people who took that loan, who did Hercules, they called that loan, and so there was a time that Bind went bankrupt, and we then ended up selling it to Pfizer. Nonetheless, the technology, even that the Bind technology, if we could go to the next slide, ended up, which are nanoparticles. All these are nanoparticles that we've done with PEG, sometimes with polymers, sometimes with lipids, but they're in clinical trials for aurora kinase inhibitors, for different anti-cancer drugs, and for gout and other diseases with Selecta. Now, I'm going to talk about, like I say, you could use lipids too, and I thought I'd go back in time and tell you a little bit about that. Liposomes, and that's a form of a lipid nanoparticle. If we could go to the next slide, liposomes were originally developed by Alec Bangham in the 1960s. And then there were lots of advances over the next 20 years by Gregory Grigoriadis, Dimitri Papadopoulos, Frank Zoka, and others. And then there was great work done by Rudy Giuliano, Chessie Barinholtz, and that would lead to new therapies. And I thought I'd show you a couple of those. This slide just shows Doxil, which is a treatment for ovarian cancer. And then also on the next slide, all these ended up being pegylated. Is Ambisome, that's for amphotericin B. And the next slide just shows that that's an approved treatment for cryptococcal meningitis. So what would happen is in the 80s and 90s, there were these different companies like Vestar and Liposome Technology Inc. and the Liposome Technology Company, and they were able to create really the first commercially used lipid nanoparticles. Now, to try to do this for DNA and RNA and things like that, I also probably should point out that generally there's like 4 components to a lipid nanoparticle, right? There's cholesterol, there's some type of structural lipid like phosphatidylcholine, there's the pegylated lipid, which I was based on some of the work we talked about earlier, and finally, usually there's an ionic lipid or an ionizable lipid. Some of the early work that led to that would be by Phil Feldner, and then Peter Cullis has done some wonderful work, and Dan Anderson, who was one of my postdocs and now a full professor, and we've also done some work on this, which I'll go over. So at any rate, when we started on this with Dan Anderson, we made this combinatorial lipid synthesis wish list. I should point out one of the things that we thought about at our lab at MIT was that liposomes and lipid nanoparticles, those had been around for many, many years, but still people were generally using about 40 or 50 different lipids to choose from. We thought, well, wouldn't it be great if you could have thousands? So we made this combinatorial lipid wish list. Next slide. And it's, you know, and the whole point is that you could do high throughput parallel synthesis. And we wanted to generate diverse chemical libraries, do it in as few steps as possible so that you didn't need to protect or deprotect what you made, that there'd be no catalysts, no solvent switches, no purification, and so forth. So just to show you some of what we did, next slide just shows some pictures of chemical diversity. We had very large variations in R groups. But what we would use actually was a Michael synthesis where we could take conjugate diacrylates with amines. So that was the key, that we would mix these with, and the next slide just shows some additional things that we did. We could not only change the R group, but we could change the tail length and the number of tails. You see 5, 4, and 3 tails on the right. And when you do this, next slide, we literally made thousands of lipid nanoparticles. We could use and we did a lot of this in collaboration with Alnylam. I've been on the founding scientific advisory board of Alnylam, since we started, since the company was started in 2001, 2002. And Kenna Kink, who was one of my students, led a lot of this work and was first author on some of these papers. But what we showed is here is that if you used in vivo assay, we'd usually use factor 7, and you can get an effect for a month. And you can also get a dose response curve, which is shown on the next slide, that what you can do is knock down factor 7 as you go from 1.25 mg per kg to 10 mg per kg. You can get complete knockdown. And you could even theoretically treat diseases like that are shown on the next slide. Like if somebody had heart disease, you could actually knock down LDL cholesterol to a high degree. If you look at the 6.25 mg per kg, you can see that it's, like say somebody had a 300 LDL cholesterol, you'd knock it down to under 200. And it's also, when you give these, it's fully reversible, and you can get very specific liver knockdown. One of the keys was that with Alnylam is that we use targeting approaches with GalNAc on the nanoparticle. And the next slide shows that you get over and over again, every month you could get knockdown. And then what Dan and I did is we say, well, could we take the very, very best lipid nanoparticles that we had, the best we call them lipidoids because they have a little bit different structures, and then make them even better. So here what we did is we took some of our leads, and if we go to the next slide, what we did is now use an epoxide chemistry to even make 150 new compounds. And again, we selected the amines from our original library. We test these in vitro, and what we find is the best ones, next slide, are 2 to 3 orders of magnitude more efficient delivery of siRNA in mice. And you can see this, the C12-200, you see the structure is 2 to 3 orders better than the LNP01. And if you go to in vivo experiments, what you see is you try to knock down trans-thiothreatin, next slide. What you are able to do that even in primates. And I probably put 2 areas that would end up being important that we really looked at. One was, could we use less and less siRNA? And by the way, all these end up, multiple companies now sell these reagents based on what we did. There's at least 4 different companies like Sigma Aldrich, Clone Tech, StemGen, and others that sell these reagents. But what we would do over time, again, working with Alnylam, is keeping on improving and improving the knockdown. So you need less and less, material. And the next slide just shows what we would do, or what we did. It's a busy slide, but if you start out and look at the abscissa and you look at 10 mg per kg, you see what the amount of knockdown you get. But over a 10-15 year period, between our work and Alnylam, we'd keep bringing this down probably 4 or 5 orders of magnitude. You know, if you go down, you see you only need 0.001 you know, mg per kg. So at any rate, we were able to show that you could make these more and more potent. We also wanted to try to see if you could do targeting to other cell types. And James Dahlman in our lab now at Georgia Tech, if we could go to the next slide, started doing screens. In fact, this was all published in Nature Nanotechnology, where we would make nanoparticles. But here we found that we could do much better knockdown in endothelial cells. If you look at these graphs, what you see is with these murine cell lines that we would get, you know, the best target. You could screen multiple organs, but what was really exciting is here, the lung endothelial cells, the heart endothelial cells, and the renal endothelial cells actually got the greatest amount of knockdown. And then we took it further. We showed in the Nature Nanotech paper, next slide, that that you could really get very, very consistent nanoparticles, lipid nanoparticles, using microfluidics. That's the way we would make them. And in fact, Rohit Karnik, who when he was in our lab did a lot of pioneering work on that for manufacturing. He's now professor in mechanical engineering. But the point is that they're highly reproducible. They're very stable over 40 days. And then if we go to the next slide, what we see is that 7C1, when you made the lipid out of that reduces endothelial cell mRNA at doses 100 times lower than any other technology. And you'd get silencing for 3 weeks, but you can get 50% silencing with 0.017 mg per kg. And then if we go to the next slide, what you see is that, again, that 7C1 preferentially targets endothelial cells. You see that on the first slide with a very high distribution. And yet here, which is interesting, is you get no hepatocyte silencing, no immune protein silencing, whereas you get robust, if you go to the bottom left slide, robust endothelial cell protein silencing. So this was all very exciting, that you could find now lipid particles that would not target the liver and would target another cell type. So I thought I'd talk a little bit about commercial things. I mentioned BIND before, where we use nanoparticles, and that didn't ultimately get the best result, though I hope will still lead to you know, now in the hands of Selecta and Pfizer and AstraZeneca, two good products. But I want to tell you an example where the nanoparticles that a company I helped start did make a big difference. Actually, Alnylam is one of them, but the one I want to talk about is shown on the next slide, which is Moderna. And this was started by myself, Nubar Fayan, Derek Rossi, and Ken Chen. And basically, as you all know, there's the central dogma of molecular biology that DNA makes, RNA makes protein. And protein therapeutics, of course, have become huge, but it takes a long time to make, right? Years and lots of purification. If you have messenger RNA, then you could just inject that into the body and it will make the protein. So we started this, but you absolutely need the delivery system. You need the nanoparticles. So we put them in, and lots of people at the company like Kerry Bonato and others have done a great job on that. But I'll just show you the Moderna pipeline. The next slide shows you just some of the things that Moderna has done. There's actually 14 in different products in clinical trials, different vaccines, different antibodies. The next slide just continues these. And you know, we're doing intracellular therapeutics, and that's another thing that the nanoparticle mRNAs allow you to do. And then different cancer therapies and regenerative therapies. Now, of course, the thing that's gotten the most attention and also the most criticism, unfortunately, in the beginning when we started to do this, people were enormously skeptical of Moderna. I mean, the stock analysts and and the newspapers and lots of scientists kept telling us over and over again how Moderna was going to fail, how it was like Theranos. I mean, there's all kinds of headlines. I'll just show you one headline, which is not very long ago when we started clinical trials. This is the Boston Globe, our local paper, talking to various experts. And this is May of the year 2000. And all these people would tell the Boston Globe, this is the headline, this is with me on the cover or on the front page, this is not how you do science, which was very discouraging. Nonetheless, what's happened as a consequence of the nanoparticles, first developed by Alnylam, shown on the next slide, that would ultimately lead to Onpattro, and by Moderna, which is shown on the next slide. This is it led to a vaccine that maybe some of you listening have taken, but it's basically been used in countries all over the world. Hundreds of millions of people have taken it. And basically, Pfizer and BioNTech have also developed a very good vaccine, again, based on nanoparticles with messenger RNA. And there are companies like Novavax also using nanoparticles to deliver vaccines. Here, I think I felt we were incredibly fortunate at Moderna to have had a wonderful CEO, which, by the way, is one of the keys for commercial success. This is Stefan Bunsell, but also lots of other wonderful people like Stephen Hogue and Melissa Moore and others who have just done a remarkable job, and Juan Andres who's done a wonderful job in manufacturing. So one other thing that I want to mention is like could you actually develop even better ways to deliver things to cells? And if we could go to the next slide, and what we could do is there are different ways that people have used, like electroporation, cell penetrating peptides, nanoparticles. If we could hit the button to go to show some of the drawbacks, next slide. So they all cause problems though, right? Damaging to the payload, chemical modification. Next slide. So they cause these problems. So I want to tell you a story. Actually, this is a story that was Klaus Jensen and I wrote a grant. We don't need to start the video yet. So we wrote a grant to the NIH trying to understand the science and engineering of how you insert things into cells. And we had this great student, Armand Shari, and he was looking at putting a gene gun. You know, and Klaus is an expert on microfluidics. So we had a microfluidic chamber that we'd shoot the cells through, and you'd notice this little constriction right underneath the gene gun. So if we start the video, can we start the video? Maybe the video is not working. It's okay if it doesn't. Notice the constriction, so that keeps going through. So one day, Armand did an interesting thing. He took away the gene gun, and shockingly, he got exactly the same result. The next slide just shows what he did. Presumably, there's nanopores that are created temporarily when you have the gene gun. So if we could just go to the next slide now. Can we go to the next slide? Next slide. Yeah. So actually, yeah. So he developed this system. We actually started a company with him as CEO. Now the company has over 100 people that are in clinical trials with this. But you could deliver, like, this was originally 1 million cells per second. Now they can do 10 billion per second. And this is next slide just shows you a picture from a paper we published in PNAS, where we took the Yamanaka factors that can reprogram fibroblasts. And if you compare conventional methods, like cell penetrating peptides, you get 2 colonies. Nucleofection, that's actually the gold standard. That's electroporation, gets 11. The cell squeeze, we got 150. So it was the most effective. And interestingly, it was also the safest. You got the highest cell survival. And what Armin and Howard Bernstein and others at Squeeze, the company, have shown is that if we go to the next slide, that if you looked at the transcriptome, I'm sorry, before that, I was going to just, yeah, I should have pointed out that in 2014, Scientific American picked what they felt were the top 10 world-changing ideas. They picked CRISPR-Cas9 first and squeeze second. And then what I was just going to show you is sometimes I make the joke that they got the order mixed up, but I think they did pretty well. But at any rate, as I was going to say, that not only has this been safer, but I'm sorry, more effective, but it's also safer. You get higher cell survival. And if you look at the transcriptome, which is the messenger RNA profile, if you look at the control and compare it to the squeeze system, it's there's much less change than if you use electroporation. And this is even mild electroporation, which is the gold standard. And this is how electroporation is how people are making CAR T cells and other things. So the squeeze method, which we're working on with Roche and other companies and by ourselves at Squeeze, I think could be a potentially hopefully much safer and more effective way. We've actually done this with 30 different cell types. So I thought I'd just show you one final slide on future challenges, and obviously there are many if you think about drug delivery, but I think large-scale manufacturing, drug targeting, which I mentioned, improved shelf life, and stability. You only need to look at what's happened with COVID to realize this. And finally, going through physiological barriers. Could you deliver drugs through the blood-brain barrier, for example? So I think there's lots of wonderful challenges ahead, but I think that nanotechnology has really made an enormous impact. In my feeling, it's changed the world. Without nanotechnology, you would not have COVID vaccines. And I think the future of the companies doing things with, drug delivery in these areas like BioNTech, Moderna, Alnylam, I think they're gonna, of course, I'm prejudiced because I'm involved in some of them, but I think it's gonna change the world even more. Thanks so much. I'll be happy to try to answer any questions.
Thank you very much. Can you hear me? Am I back on? Yes, fantastic. This was a really fantastic and enlightening talk, and really inspiring. Also hearing about the challenges that you had to go through during your career. So yeah, we have a few questions. So I'll read the first one for you. You've been asked, what is your opinion regarding a potential shift in the applications of nanoparticles in the future? Do you think more attention will be given to neurodegenerative diseases and less to cancer?
I think it's a good question. I think it's going to be across the board. I think all these diseases are important. Part of the issue with both neurodegenerative diseases and cancer isn't, I wish it was just delivery. Delivery is certainly one huge area, and I think it can affect and has both those diseases. But we also need to understand the biology better, for both neurodegenerative diseases and cancer.
So a second question is, does the extensive use of pegylated materials raise a concern in terms of immunogenicity? Do you expect a diminishing in PEG use or not for creating new delivery systems?
I think that PEG has been very, very useful, and I think it probably will continue to be, but there are other areas, other groups working on things like this. In fact, Xiaoyi Zhang, who's a professor at Cornell, then I've helped him a little bit, has created new polymers like, zwitterionic polymers, and I think that that's also a very, very exciting, important area. So I think there's certainly an opportunity to come up with other molecules that can be used for this kind of purpose.
Yeah, then there was actually another question related to this and related to infusion reactions and the side effects of the COVID vaccine related to PEG, and if there are PEG substitutes that have been proposed.
Yeah, well, I think that I don't know that. It sort of goes to what I said, that I think that like Xiaoyi Zhang has done a lot of work on this, and there's another group. Milt Harris has done some work on this. So I think that there are different molecules. Xiaoyi's published quite a few papers in different journals showing very good effects of some of his zwitterionic polymers, one of which got approved as a coating for different cardiovascular devices, PIC catheters.
I'm just scrolling through. So another question that we received is, what do you think about nanotoxicology due to common use of nanoparticles?
Well, I think it depends how you look at it. I think that, you know, safety is always important. I think the biggest issue is what the nanoparticles are made out of. You know, I think if you make them out of something that's bad, that's going to be bad. If you make them out of things that are safe, I think they'll most likely be fine. But it could be amplified. You know, if something is, you know, for some of the same reasons that things are positive, I could imagine if you've got nanoparticles in the air, you know, people could breathe them. I mean, you know, viruses are nanoparticles too.
Yeah, and a somewhat related question is how much cationic nanoparticles can play a role in delivering macromolecules and their side effects?
Well, I think there's no question that cationic, you know, I think that a number of groups have shown that, like Peter Cullis and others, you know, and Phil Feldner. You know, I think there's no, and then Dan Anderson and myself and others. So I think there's no question that they're useful, but I think you do want to watch out for things like complement activation and so forth in terms of side effects. I think you certainly can get side effects. So you need to screen them early.
Mm. And another question that just came in is, what do you think about incorporation of magnetic nanoparticles in drug-controlled release systems?
I think it's an exciting area. I mean, people have tried that before, including ourselves. We've actually put, going back into the 90s, I remember we put nanomagnetic particles into lipid, or magnetic particles into lipid nanoparticles. And there was really a group, Senier and Witter pioneered some of that work. I remember back in the late 70s and early 80s. The challenge is making sure that they all go to where you want. I think one of the problems is that there was some toxicity when this from the Senier Witter work, even though it was really pioneering work. Some of it went to the brain, unfortunately.
Okay. And a slightly different question to the previous ones is, what was your motivation behind establishing pharmaceutical companies, and what is your advice to young scientists regarding science and business?
So my motivation was twofold. One, I wanted to, and my students wanted to see the work we did not just be scientific papers, but actually help people. And sadly, I found out the hard way that just publishing papers wasn't enough. And in fact, and even licensing them to large companies, which I started doing into the 80s, wasn't enough either. They just, I mean, that's a whole story I can tell you, but we licensed technology to 2 giant companies in the 80s for macromolecule delivery. And I mean, it was, you know, I remember I got consulting fees and I got, you know, big grants from them, and I was very excited about that. What I was most excited about was that they were going to develop it and it would go to patients, but they just gave up too easily. And that's why I started companies, because I did it. And that gets the second reason I do it with my students, because they also wanted to see, you know, the work they did, you know, make a difference in the world. And so we kept doing it over and over again. And so, but the small companies like Alchemies, like Moderna, like Alnylam, I mean, of course, they've grown up, but they've changed the world. I mean, they have made products that affect, you know, like I said, probably billions of people. And it's also created a tremendous number of jobs. So, I mean, but that's why I did it. And still do it.
So another general question is if you think that you know, the development of the COVID vaccines, what will be the benefits for the nanomedicine field in general, and if you can expect more FDA-approved nanomedicines in the future.
Absolutely. I mean, I think that no question. I think the precedent establishes shows that they're very safe and very effective and can protect molecules that would otherwise be destroyed, important molecules like messenger RNA.
So I have actually a question myself. You told us that, you know, you've often encountered a lot of criticisms or a lot of challenges to actually pursue what you thought was something that would benefit, you know, patients, eventually patients. How did you and how do you choose which battles to fight in a way?
You fight them all. I mean, because if you write, I mean, to me, if it's something that can help do either good science or help save lives, I mean, I think you want to keep trying, whether it was the angiogenesis work or the initial drug delivery work or the Moderna stuff. I mean, all those ended up, you know, affecting hundreds of millions, if not billions of people. And I like to think they led to, you know, important science as well. So I think they're worth fighting for.
And actually, I just received a question that kind of linked into what I was thinking. So what do you think the next big, exciting research target in the field is going to be? And where do you think, that there are some specific technology that is close to be, you know, translated to to actually the real life in a way?
Well, I think there are many areas. I mean, you could look at the Moderna pipeline or the BioNTech pipeline or the Alnylam pipeline. They're all, and many other small companies or medium-sized companies, I think, or large ones. I think there's just so many. I think I'd love to see bigger impact on cancer, for example, and I think we probably will. I'm not sure exactly where it will be, whether it's personalized cancer vaccines or other things, but I think all these things are important.
Fantastic. Well, thank you very much. Really, it was really a pleasure to have you here, and we were really thrilled that you accepted to talk with us and to give a seminar for our anniversary. So it was really great to hear your perspective and to hear your story, really inspiring and enlightening. And thanks to everyone that followed us today. I have one announcement to make, actually. So for the next series of webinars, so we're going to have like 2 webinars every Wednesday. So for the next series, you will receive a link for the next seminar, in the email that you receive after the webinars has finished. So thank you very much again to everyone. It was a pleasure, and thanks again to the speakers, and hopefully you'll join us next week again for another really exciting session of speakers. Thank you very much. Bye.